Hello, my name is Ian Campbell, and as part of my undergraduate research in Dr. Kim's lab, I am presenting the role of ADP-ribosylation and its reversal by ARH1 in the DNA damage response. Beginning with a refresher of DNA, the central dogma states that DNA is transcribed into RNA, which is then translated into proteins, essentially meaning that all proteins in and around us have a DNA foundation. Consequentially, damage to DNA gets transcribed into damaged RNA and damaged proteins. Many harmful conditions result from this chain of events, one of them being cancer. Cells are naturally exposed to damage as the cells get older or when toxins are introduced. Even still, cells are still able to maintain their own integrity. One of the ways they do this is through repairing the damage itself. This is just one pathway of many important to regulating DNA damage. A key feature to note is the post-translational modification, known as ribosylation. This is the addition of one or more ADP-ribose units to proteins. In the case of PARP, several long chains of ADPR are bound once it reaches the damage site. When it is no longer needed, hydrolases, another protein, remove the ADPR groups from it so that it can locate other sites of DNA damage in the cell. ARH1 is one such hydrolase. However, instead of removing long branching chains, it removes only single additions. Before taking an in-depth look into ARH1, purification of each important protein for the reaction is necessary to eliminate external factors. And the first step before purification of any protein is transformation. This requires a bacterial plasmid, which is a circular string of DNA that contains a synthetic gene for the protein we want to multiply. At least one other region contains some antibacterial resistance so that when we introduce an antibacterial agent, only the bacteria cells that have the plasmid survive, allowing us to isolate only the cells that will make our protein. Plasmids are easy to introduce into a bacterial culture, needing only a heat shock. After inducing expression overnight, the bacteria are then centrifuged and combined with one another into a clump of cells. This solution is resuspended in a buffer solution and ready for purification. This begins with nickel affinity chromatography. The resuspended cells are pumped through the column on the right from top to bottom. Our target protein, the one in purple, has what's known as a histidine tag, which is a series of 6 histidine amino acids next to each other. They conform in such a special way that allows them to bind to nickel, the green diamond, as the solution passes through the column. At the bottom, another tube collects a fraction of the solution being pumped. The column is then washed with a different buffer to remove as much of the cell debris as possible. And with the help of an elution buffer, the target protein is pushed out of the column last. The next two steps are similar to each other and use the same instrument, an FPLC, shown on the right. The goal is still to separate proteins in ion exchange chromatography, as it was in the nickel affinity chromatography. Only this time, instead of the histidine tag, it separates what is remaining by net charge. This process takes much longer and is more exact, this time collecting 70 fractions. The instrument's detector lets us know based on a peak which fractions contain our target protein. Size exclusion, or separation by size, is the last step. The graph at the bottom is comparable to data that we receive in ion exchange that I was referencing. The fractions number at the top, A36 through A41, indicates fractions 36 through 41. Using gel electrophoresis of fractions 31 and 40, we can confirm, based on the known molecular weight of ARH1, that what we have left in those fractions is ARH1, and the protein has been purified successfully. Using purified proteins, we can perform experiments like this Western blot, which can identify protein contents of a solution. We can see the effects of ARH1 on ubiquitin mono-ADPR. On the right, only 2 lanes detect ADPR. This is due to the mixtures that were prepared. Of note is that the middle band on the right is thicker than the rightmost band, and since the only difference between the two is the addition of ARH1 on the right, some ADPR must have been removed by ARH1. A test such as this, and the future procedures for a more in-depth analysis of ARH1, require protein purification for any clear results, especially if the goal is to create an interaction model of ARH1. Impurities would only hinder our further understanding. Thank you.